Hi everyone, in this video we're going to use artificial cells to practice scientific experimentation and data analysis and also support our understanding of diffusion and osmosis. So let's get started. You recall our brief introduction in class on how to make an artificial cell. Essentially, you're going to cut a 6-inch strip of dialysis tubing. That's that flat, dry plastic material that needs to be put into water so that it can be opened up into a tube shape. Okay, so we like to make them a little bit longer than the actual cell because we're going to seal up the ends with clamps. So here's your artificial cell. I just sort of draw them as little circles. Here I'm showing you what it'll actually look like with the clamps on it, but I will, in future pictures, not draw the clamps. So we'll take this cell to the scale, and we're going to mass this cell. So here you see our normal high school biology scale, where you'd read the mass of the object here in grams, and our cell here is placed on a weighboat. So we're going to use mass as our dependent variable, more specifically percent mass change, which I'll talk about in a moment. So we're going to see how mass changes in artificial cells depending on the solution that it's in. So for your experimental setup, we'll give you beakers where you can put in a certain volume of solution and where you can add your artificial cell into that solution, so your artificial cell will be surrounded by a solution. And we'll make sure that the solution is a different concentration of sucrose than the solution inside of your artificial cell. Okay, so the time that it'll take for this to occur is about an hour. So you can get experimental data with artificial cells in about a class period. So we will construct our cells quickly at the beginning, wait an hour, and then we'll mass them first and then wait an hour and get a mass afterwards to compare percent mass change. So the really important part is that after your cell is submerged in a solution, that you take another mass to get that final mass to calculate percent mass change. So what you're going to want to ask is the following questions. Did your mass increase? Did your mass decrease? Or did it stay the same? Okay, so we're going to, instead of just looking at mass change, which is sort of an arbitrary number because the cells are going to be different masses due to how we construct them. One might be 10, one might be 11. So we're going to normalize the numbers by converting this to percent mass change. To do that, we take the final mass and subtract the initial mass from it. We divide that number by the initial mass. Then we multiply that number by 100 to get a percentage. For example, if your initial mass is 9.2 grams and after 1 hour's time, your final mass is 8.2 grams, that means you've lost 1 gram. When we divide that by the initial mass of 9.2 grams, you'll get this number, which multiplied by 100 will give us 10.8%. Now that is 10.8% decrease because our mass has gone down. And I say this just to remind you that the mass of these artificial cells can increase or decrease depending on where osmosis is going to occur. So osmosis might bring water into the cell, or it might bring water out of the cell. So let's start the lab. You're going to have 3 mystery solutions. I've colored them A, B, and C in different colors. So unknown mystery solution A will be red, B will be blue, and C will be yellow. Now we know that we have 3 solutions of unknown concentration. They could be either 30%, 10%, or 2%. So this is fun. This was what makes it kind of a challenge. You know that these are the 3 solution concentrations, but you don't know which is which. So here's the parameters of the setup. Okay, so we have to sort of identify the rules of the game. So for artificial membrane, so for this dialysis tubing, very small molecules can pass from one side to the other. So dye and water will be able to diffuse, so it can cross. Now, sucrose, on the other hand, which is a disaccharide, is too big to diffuse out of the bag. So you're not going to see sucrose go from the bag or the solution in opposite directions. So because it's a disaccharide, it's too big to move. Monosaccharides like glucose can move, but we're not working with that molecule for this reason. We want to keep it either in the bag or out of the bag to eliminate diffusion of the solute as a possibility in this experiment. We just want to track osmosis, which will contribute to mass gain or loss in the bag because water is going to be the most abundant molecule here. Okay, so we're going to calculate this as percent mass change. A reminder about the types of solutions that we're working with, the tonicities, if you will. So if you're in a hypertonic solution, that means the solution outside of the cell relative to the inside of the cell has higher concentrations of solute and lower amounts of water. Conversely, the inside of the cell has lower solute but more water. Okay, so that's the description of a hypertonic solution. Isotonic solutions have the same amounts of solute and water, whereas hypotonic solutions surrounding the cell have lower amounts of solute but higher amounts of water. So in this hypertonic solution in general, water will leave, resulting in mass decrease. And in a hypotonic solution, water will enter, resulting in a mass increase. So let's set up this virtual experiment. To do this, I want you to think of all the possible combinations of solutions that you could create in an artificial lab, okay? So if we were here doing the lab for real instead of virtually, what sort of experiments would you set up? What would you put in your artificial cell, for example? What would you surround it with? An example of a useful combination would be having A inside the bag, but B surrounding the bag, okay? So submerging a cell with A in it, in a solution that has, which is solution B, okay? So how many combinations can you think of? Well, here's what comes to mind for me. I want to put as many possible combinations as possible. That's going to give me the most amount of data, which I can then use to figure out what these mystery solutions are percentage-wise. So for this first set of experiments, I'm going to make 2 artificial cells that have A solution inside and submerge them in the other solution. So for example, I'll put A inside of a B solution, A inside of a C solution. Likewise, I'm going to make B containing cells that are submerged in A and are submerged in C. Now to complete all the combinations, I want to make cells that have C solution inside and submerge that cell into solution A, and in a separate experiment, submerge it in solution B. Now down here, I have 2 possible controls that we can do. There are a lot of other controls that we can do, but I find this control useful for experimental error. Because we're constructing so many cells, we can mess up and mix up solutions, and this kind of controls for that. So if I have a cell that contains B solution surrounded by B solution, I shouldn't see that much of a percent mass change. Now, why is that? Well, this is an isotonic setup. In isotonic setups, there should be an equal amount of osmosis in both directions. So water should go into the cell, and just as readily it should come out of the cell. Same thing for our control over here with A inside the bag and A in the solution surrounding the bag. As always, it's useful to have a data table to keep track of the data that you're obtaining from your experiments. So here's a data table that we could use, and one, in fact, that you'll see in your Google Doc. Okay, so for this column, I'm tracking what's on the inside of the cell and what's on the outside of the cell. And for that particular experiment, I'm giving you here space to keep track of what your initial mass was and what your final mass is after 1 hour. I'm then giving you a space here to keep track of your percent mass change. So after you've calculated your percent mass change, put that number here. We will need that data to perform an analysis that determines the mystery solution percentages. how much sucrose they have concentrated. So here's your experimental data for this virtual lab. So I'm giving you here some virtual data that we see experimentally when I submerge a cell that contains A in a solution in a flask of B or a beaker of B. So that cell, its initial mass is 10.2 grams, and its final mass is 8.8. So go ahead and put that into your data table so that you can use that to calculate your percent mass change. Over here, I have A inside the cell, and C solution is surrounding it. That gives us an initial mass of 9.1 and a final mass of 10.2. Please add that to your data table. Here are the B cell experiments. Here B cells are put into A solution. Here a B cell is put into C solution. In this particular experiment with A inside and B inside and A outside, the initial mass was 11 grams and the final is 12.4. Here, the cell containing B put into solution C, the initial mass was 10 grams and the final is 12.7. Here's the C cell experiments. The cell containing C submerged in A, its initial mass was 10.1 grams. Its final was 8.8. Here, this cell submerged in B, its initial mass is 11.2 and its final is 7.7. Here's our control data. So in this experiment, initial mass 11.1, final 11.2. Initial for B cell in B solution, 10.8 is the initial, and the final is 10.6. Okay, so that's the end of this virtual lab. I would like for you now to calculate your data as if we were doing a real experiment and use that data to predict what the actual percentages of each solution is. So I'd like to see how you're thinking about this. I'd like to see you take that experimental data and analyze it in order to draw an accurate scientific conclusion, and we will discuss this in class next time.